Hi everyone, we are Group 3, comprising of Jinxiang, Riz, Karina, Celian, Sinsio, and Wenxuan. Today, we'll be presenting the rationale and design of our study, which explores the effect of oxygen supplementation on the soil microbiome. Many new plant owners can relate to the anxiety of bringing home a new potted seedling or transferring a cutting into soil. The successful rooting of the plant is critical for its survival. Yet in many cases, seedlings and cuttings fail to thrive, especially in the hands of inexperienced plant owners. To increase their chances of success in rooting, we often turn to rooting hormones, which contain auxin as their active ingredient. Auxin is a class of phytohormones that plays a central role in regulating plant growth and development, including phototropism, apical dominance, and root formation. The primary naturally occurring auxin is indole-3 acetic acid, which is mainly synthesized in the shoot apex. Its synthetic analog, 1-naphthalene acetic acid, is a common active ingredient in fertilizers and commercial rooting formulas to promote plant propagation. At a molecular level, auxin mediates the degradation of OXIAA transcriptional repressors via the TIR1 SCFE3 ubiquitin ligase complex, therefore functioning as a double negative repression activator of auxin response transcription factors. These ARFs are now available to promote the expression of auxin-induced genes responsible for a wide variety of plant developmental processes. It has become a common horticultural practice to apply auxin supplements to encourage rooting and growth in plant cuttings. Apart from its essential role in plant development, auxin also serves as a viable carbon source for various soil microbes in the rhizosphere. This suggests that auxin supplementation could influence microbial growth and microbiome composition. However, the impact of auxin supplementation on the soil microbiome remains poorly characterized. This gap in knowledge is especially concerning given the widespread agricultural use of auxin-based products, particularly herbicides that apply high concentrations of auxin derivatives to suppress weed growth. In large-scale farming, such practices could unintentionally alter soil microbiomes, potentially compromising the long-term fertility of farmland.
Building on that, let's move on to our specific research question. How does auxin influence the diversity and composition of the soil microbiome? We hypothesize that adding auxin to soil will enrich microbial diversity, particularly bacteria that support nutrient uptake in plants. At the same time, we expect an increase in phytopathogenic species that exploit auxin-stimulated root growth to infect plants. To guide our design, we reviewed studies on auxin microbe interactions. Abu Zara et al. Found that optimal auxin concentrations for plant rooting ranges between 1,000 and 3,000 ppm, while higher doses, such as 5,000 ppm, inhibited growth in rosemary. Matilla et al. Showed that increasing IAA levels suppress bacterial antibiotic production. The antibiosis halo dissipated completely at 400 micromolar, demonstrating that high auxin can inhibit microbial metabolism. Such inhibition is undesirable for our study, as it could reduce microbial diversity and confound our results. Therefore, we carefully selected an auxin concentration of 10 mg per ml, high enough to promote root growth but low enough to avoid bacteriostatic effects. In our study, we used money plants because it's inexpensive, common as a houseplant, and young plants are ideal for rooting studies. Auxin in the form of IAA and NAA was used. The auxin mix was suspended in water to make up 10 mg per ml watering solution, and we then measured 100 ml of this watering solution and watered onto the soil directly for all 6 plant samples. Finally, we let the plants to grow under indirect sunlight for 7 days before obtaining soil samples for soil microbiome assessment.
I will now talk about our sampling technique. For each plant, about 1.5 grams of soil was collected from near the roots before and after auxin treatment. After collection, the samples were immediately frozen at minus 80 degrees Celsius to preserve their microbial composition. This sampling technique has several key advantages. By collecting soil close to the roots, we focus on microbes most affected by auxin-induced root growth. The rhizosphere is also rich in nutrients released by the plant, such as sugars, amino acids, and organic acids, which support active microbial communities. Auxin can change the amount and type of these root exudates, so sampling here helps us capture those shifts more directly. Next, by taking both pre and post samples, we can compare how the microbial composition changes over time. The pre sample gives us the baseline, and the post samples show the effect of auxin while reducing differences between plants since each plant serves as its own comparison. Finally, freezing the samples immediately at minus 80 stops all microbial activity, keeping the community exactly as it was at collection. This also prevents DNA and RNA degradation, which is crucial for accurate sequencing and downstream microbial analysis.
Our study had several key limitations. First, the auxin treatment was inconsistent. It was difficult to control the exact concentration that reached different root zones. meaning some of the plants may have received a higher or lower dose than intended. We also faced unforeseen disturbances. A snail, Turbo, was found in one of our samples, which could have altered the soil microbiome. Furthermore, the plant's location near the lab's hand washing and personal item storage area with high movement risked contaminating the plants with external microbes. A major limitation was scale. We only used 6 plant replicates, which reduced our statistical power in a field known for high variability. The soil sample volume was also very small, raising the risk of missing the full microbiodiversity. The study's short duration and single treatment mean we can only see short-term microbial responses and potentially missing any long-term or cumulative effects. Finally, there were inconsistencies in our starting material and sampling methods. We don't know if all plants started with the same soil batch, which could have led to different additives. We also sampled from different locations in the pot each time, introducing spatial variability. Additionally, we lacked controls for key environmental variables. For instance, we observed unexplained leaf yellowing in one of the samples. For future studies, microbiome profile comparisons could be carried out based on the 16S RNA sequencing result for plants with and without the yellowing effect to determine if the yellowing is caused by intrinsic or extrinsic factors. Sunlight exposure and soil moisture content were not measured due to the time and budget constraints, leaving potential environmental influences unaccounted for. However, a pyranometer and a moisture meter could be procured for these measurements in the future.
Factors that could not be controlled or measured. Number one, the amount of indirect sunlight received by the plants at the lab window at varying times of the day. Moreover, the lack of space resulted in plants being confined to a tray, which might have resulted in an uneven amount of sunlight distribution for the plants behind. Secondly, each plant might have differences in the soil and moisture levels, and because additional soil was not bought due to budget constraints, we are unable to control the moisture levels. Number three, central building temperature conditions might have hampered the plant's growth due to suboptimal temperature or unacclimatized plants. Number four, significance of these factors for sunlight is that different sunlight exposure may cause different growth rates due to different photosynthesis rates, affecting any microbe-plant symbiotic relation present at the roots. For potted soil, the starting microbial composition may vary between the plants. Moreover, varying moisture levels might have altered microbial diversity and abundance. For temperature, similar to sunlight, a lower temperature means slower growth, which may alter the diversity and abundance of microbial species found at the roots. pH changes by auxin makes it difficult to determine if the microbial diversity changes are due to pH or auxin treatment or both factors. Lastly, for short intervention length, changes in microbial composition might not be seen and there is no observation of long-term effects.
Without resource constraints, we can put the plant in a greenhouse with control parameters such as consistent light source. The greenhouse would also solve the issue of temperature fluctuation as temperature and humidity could be set. The issue of inconsistent moisture level could be resolved by using a soil moisture probe after watering the plant to ensure consistent moisture level in all samples. Lastly, we could measure pH of the soil before and after intervention to determine if the pH change is due to the intervention. As the ideal pH is 6.1 to 6.5, we could neutralize the soil with basic material if the plant after intervention is too acidic.